Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Tony Galenza, and I'm a postdoc at Stanford University, and we'll be moderating today with Olya Yarychkivska, a postdoc at the Rockefeller University. We're excited to highlight the work of our outstanding postdoctoral members. Today, Weiyi Qian from New York University Grossman School of Medicine and Robyn Allen from the University of Kentucky will share their research. Each speaker will give a 20-minute talk, followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. And with that, it's my pleasure to introduce our first speaker, Dr. Weiyi Qian, a postdoc fellow in the lab of Dr. Holger Knaut at the NYU Grossman School of Medicine. Weiyi earned his PhD in mechanical engineering from NYU in the lab of Weiyi Qian, where he engineered nanotopographic cues to regulate mesenchymal stem cell osteogenesis and developed integrated microsystems to investigate single-cell mechanical phenotypes. During his postdoc, Weiyi's research has focused on uncovering the physical, molecular, and cellular mechanisms that drive collective cell migration and tissue morphogenesis during embryonic development. As part of this work, he published a beautiful paper in Current Biology showing that pulses of Rho A signaling stimulate actin polymerization and flow in protrusions to drive collective cell migration. Weiyi has also been granted multiple competitive awards, including an American Heart Association Fellowship and an NIH K99 Award. In today's talk, Weiyi will share his current work on how tissues split through cell-cell and cell matrix adhesions. Please join me in welcoming Weiyi.
All right. Thank you, Tony, for the kind introduction. And also thank you, SDB, for this opportunity to present my postdoc work. So today I'm going to share with you a story about how a living tissue split into two to form a functional organ unit, and how this process is being mediated by the adhesion between cells and also between cells and the matrix. So tissue splitting is actually very common during development. And the video you see now is actually the formation of the early heart tube through the splitting of the endocardium cells. Another example of splitting is the formation of the esophagus and trachea through the tracheoesophageal separations. The video you see now is actually a Z-stack from the shared foregut to the separated channels that formed later by splitting of the shared foregut. Starting of tissue splitting in vitro is actually challenging. One of the big challenges is that you need to have a model that you can really perform high quality live imaging so that you can look into the details and dynamics at the subcellular and cellular activities. So that's the first requirement. Second, you should be able to do tissue-specific perturbations so that you can remove molecular players that you think is playing a role in this process. At the same time, split tissue required forces to be generated and transmitted and received by the cells. We should be able to do the biophysical measurement in this process. So this is presenting challenges to study tissue splitting in vivo. So what I'm going to do today is I'm going to briefly introduce you the model I'm using, and then I'm going to talk about how cells in this tissue that I'm working on start to repolymerize their protrusions to pull on each other to generate tension, and then how this tension splits the tissue. At the same time, I'm going to talk about how this tissue need to adhere to the underlying substrate so that I can split the tissue. So the model I'm using is the zebrafish letter line system. So the letter line is a sensory system for the fish to detect the water flow in their surroundings. What you see here on the screen is a zebrafish embryo at around 19-hour post-fertilization. Here's the head and here's the tail. And at this time point, a class of cells is born directly behind the ear of the fish embryo, which is about 140 cells. And in the next 20 hours, this tissue will start to migrate along the body from the ear to the tail. And during this migration, this tissue will deposit a series class of cells from its real, which later will differentiate to form a sensory organ called the neuromast. The neuromast can extend their cilia to the outside to detect water flow. Here's an example of this migrating and depositing process. You can see we can really capture the dynamics of this process. And the great advantage of using this model is that this tissue is migrating directly underneath the skin of the zebrafish embryo. So give us really great optical accessibility to probe the dynamics of this splitting process. So now I'm going to tell you why these cells in the real start to bud off from the tissue. And that is because cells in the real reverse their protrusion direction and pull them on the cells in the front. So to do that, we need to do transplantation to label single cells in the tissue so that we can look into details of the protrusion activities. This is because the cells in the tissue is overlying with each other. If we do not label single cells, we cannot look into these details. And then we can group the cells in the tissue into different groups based on their locations, and then study how the protrusion behavior different for cells at different locations. So here we label the cells with F-actin, which we can check the protrusion activities. The host embryo will be labeled for nuclei with GFP. Here, just to show you an example of a migrating tissue that with two clusters labeled with F-actin. Now, if you look at the first cluster in the front, you can see the protrusion mostly protrude forward, and you can see dramatically F-actin polymerization. Now, if you look at the cells in this cluster in the real, you can start to see that some of the cells, especially here, is protruding backwards. If we collect enough data and group the cells based on their location in the tissue, we can then quantify the protrusion activities. So you can see cells in the back start to protruding backwards, while cells in the front and the middle mostly protruding forward to migrating together with the tissue. You also notice that tip cells protrude pretty roundly, and that is because these tip cells need to probe their route for the tissue to migrate, so they actively protrude in different directions. So now propose a model that as the cells in the real reverse their protrusion direction, they might pull on the cells in the front, and that will build up a tension between the deposited neuromass and the migrating tissue. And we want to test this hypothesis by using a method called laser ablation. So the laser ablation can be best illustrated by pulling a rubber band. So imagine that you're pulling a rubber band and you cut in the middle, your two hands will recoil because there's tension. If you pull harder and you cut in the middle, your two hands will recoil much faster. This is the same that we're going to use for this tissue. So as I show you the video later, I hope you can focus on the two sides of cells at this ablating site. If these two structures, the cells in the real and the cells in front, is pulling on each other, when I ablate the cells in the middle, they should recoil in the opposite direction. Now I'm going to play the video. So I hope you can see that the tissue is recoil in the opposite direction. We can quantify the movement of these cells at the cutting site and quantify the velocity of recoil to add the readout of how much tension is accumulated at this site. Further, we can study the tension dynamics during this splitting process. To do that, we divide this splitting process into 4 stages. First, before the splitting starts, where the neuromass still migrating together with the tissue. And then as the tissue continues to thinning, we can divide it into further 3 stages. We can then apply a laser ablation at different locations before the splitting starts, and also during the splitting process. Imagine the cells we're cutting the tissue in the front. Because the cells in the front is migrating together, we should see less tension compared to the back. This is what we observed. Now, let's focus for the first 2 columns. The first column shows the recoil of the cells in the front of the tissue before the splitting starts. And you can see there's a significant increase of tension in the back of the tissue. As the tissue starts to split, you can see the tension continues to decrease, suggesting as the tissue splits, the building up the tension is start to dissipate by the cells in the real. How does this dissipation of tension happens? One possible way to do that is if we apply tension to a tissue, and the tissue can change shape through the cell position change. This cell position change can use to dissipate tensions, so they don't accumulate it. And we try to test whether this is also true in the primordial. And for that, we measure the mean square relative displacement. which is a parameter to quantify how much a cell changes location relative to its neighbors. Before I play the video, I want you to aware that the color bar shows how the magnitude of the position change. If you see a red color, meaning that the cells is changing neighbor much more than the cells showing a blue color. So as I play the video, I hope you can start to appreciate that cells in the back start to show much more position change compared to cells in front. So suggesting that these cells in the real are actively changing positions so that they can dissipate the tension because of building up tension due to the reverse of the protrusion directions. Now, because cells are connecting to each other through cell adhesions, one of the adhesion molecule is E-cadherin. E-cadherin includes the extracellular domain and the intracellular domain. Intracellular domain is connected to cytoskeleton through alpha-catenin, which is here denoted as a gene name CTNA1. So if we remove the adhesion between cells, they should be more freely to move around and should be able to dissipate tension much efficient than Y-type embryos. So to do that, we take advantage of a alpha-catenin knock-in line and label the alpha-catenin with citrate. Then we can use a degrant system to deplete this citrate-labeled alpha-catenin, which we developed in the lab called Zgrat. How we do that is we take the alpha-catenin citrate knock-in line, we tissue specifically express the Zgrat, which is a degrant system in this tissue. If we express it, this tissue will be, the alpha-catenin will be depleted from this tissue. So here I'm going to show you an example of how this tissue looks like if we remove the adhesion between cells. So the top panel is the Y-type embryo, and the bottom is the one with the alpha-catenin depleted. And you can see compared to the Y-type, the tissue in the bottom shows a more fluidized behaviors. Cells kind of move randomly and cannot form cohesive clusters as deposited neuromass. We can then track each cell's behavior to see whether the cells in the alpha-catenin depleted embryo really show a larger position change compared to the Y-type. So we developed the automatic tracking system to automatically track the cell position in this process. We can then do the same analysis, which I showed you before in the Y-type, which is called MSRD. If I play the video, I hope you can see that cells in the real of the alpha-catenin depleted embryos shows a much larger increase of position change compared to the Y-type, suggesting they are being able to move freely and also being efficiently to dissipate tensions. This is quantified data. As you can see, compared to the Y-type, embryos with alpha-catenin depleted shows a much more ability to change neighbors compared to the Y-type. This is a behavior. This is a movement. They can easily move compared to the Y-type. And then how about the tension in these conditions? This is a complicated graph, but let's go through each step. So record the tension trend I showed you in the Y-type embryos. The tension, which is denoted in the white arrow bar. As you can see, tension is in the Y-type, tension is building up, then dissipated during the splitting. Now let's look at the arrow bar with the magenta color, which is the tissue that depleted alpha-E-catenin. You can see in this group of tissues, tension cannot be built up. However, they can dissipate tension much more efficiently than the Y-type tissues, suggesting cell adhesion play a role for building up tension, and also without that, cells can efficiently dissipate tension and dissociate without efficient splitting. So here's the model for now that we now see a tension between the deposited neuromass and the migrating primordians. The next question is, if the migrating primordial is pulling on the neuromass, does the neuromass need to adhere to the underlying substrate to resist pulling? Right? Before answering this question, there's some background about this tissue. This tissue is actually migrated on top of a basal membrane. This is the EM picture showing the cross-section of the embryo with the migrating tissue. If we zoom in, you can see this pseudo-colored part, which is the primordial, is actually migrated on top of base membrane. So to answer that question, we can then label the component in the base membrane One of the components is laminin gamma 1, and we can label it with GFP. We then place a bleach dot or pattern along the root of the migrating tissue, which is showing as magenta. Now, as this tissue migrates on top of this pattern, we can then track the movement of those bleached dots to see how this tissue is deformed the underlying substrate to see whether the neural mass need to adhere to a substrate to resist pulling. If we zoom in the front, we should expect to see these dots move backwards. This is the same as when you walk on a beach, you kind of push the sand underneath your foot backwards. Now in the back, if this class of tissue need to resist pulling of the cells, they should pull these dots forward. We want to test whether this is true in this model. So this experiment we did, we bleached a pattern of dots in the laminin gamma 1 GFP fish line, and as this tissue is migrating on top of it, it will deform the substrate, and we can track the movement of these dots. I have to say this is still a work in progress. We're still trying to develop a computational model to quantitatively detect the flow movement of these dots. But for now, I can show you preliminary observations of how these dots move by simply generate a chemograph. So here's a video that the neural mass migrate on top of this bleached dot. And then we can get a chemograph along this of the dots along the road of the migration. So as you can see in this chemograph, on the left, you can see this dot is being pulled towards the right, where on the right, this dot is being pulled towards the left, suggesting that the neural mass really need to resist the pulling of the migration primordial. So the next question we're interested in is, if the tissue need to adhere to the underlying substrate to resist pulling, does it adhere to a substrate through integrins? So integrins is a transmembrane receptor that recognizes the actual cell matrix and connects it to the cytoskeleton. It requires its alpha subunit and beta subunit to form a heterodimer to recognize specific component in the actual cellular matrix. Here is just a graph showing part of the heterodimer that the integrin can form in the tissues. So the strategy we try to use is directly mutant certain integrin in the tissue and then see what happened to the splitting event. If we remove alpha V integrin, we did not see any type actually. The migration is normal. The neural mass can be depleted efficiently. Now, if we remove the integrin beta 1 in the tissue, we start to see something happening. Here is a Y-type embryo. You can see this bright dot, which has a deposited neural mass, and the primordial is migrating all the way to the end. If we remove integrin beta 1b, get an integrin 1b mutant, you can see the primordial is now cannot migrate to the tip, and the distance between the clusters of the deposit neural mass get closer, but you can still see some minor structures. If you further remove one more copy of integrin beta 1 in this condition, you can see this deposit neural mass are almost gone, suggesting that the phenotype is become severe without enough integrin activities. Unfortunately, we cannot study the phenotype for the integrin beta 1a and beta 1b mutant. This is because the fish cannot grow a body trunk for the tissue to migrate on it. Now, if we look into detail and use the second neural mass as example, you can see compared to the Y type, removing one copy of beta 1a and two copies of beta 1b, the neural mass become much more smaller. This is the same for the third neural mass if you look into the details. So what happens is that without integrin, the cells cannot efficiently adhere to the substrate and resist pulling, so they are being pulled away by the tissue and cannot split efficiently. So in summary, what we find in this model is for splitting a tissue, the cells in the rear of this migrating tissue will repolymerize their protrusions and pull on the cells in the front. This will create a tug of war that cells in the front and the tissue are pulling on each other. At the same time, cells in the rear need to also adhere to the underlying substrate so that they can resist the pulling to be deposited. So with that, I would like to thank all the members in the Connaught lab, including the past lab members. I would also like to thank our collaborators who helped to build up automatic tracking system and quantify the tracking stress in this process. I would thank our collaborator, Torres Vasquez Lab, and all the microscopy core facility here. Without them, we cannot do all the imaging. Thank you all the funding support, and of course, for the fish for me to work on that. With that, I'm happy to take any questions that you may have.
All right, just a reminder, put your questions in the Q&A box. Yeah, that was really beautiful, Weiyi. I'll start. I was really interested in the variability that you see in this process. How consistent is like the number of cells that get left behind and the location that they drop off?
Yeah, so there's definitely some variations even between each embryos. So the pattern of the deposit neural mass is dependent on FGF signaling actually. So this cluster of cells continue to sense FGF signaling. Once it's reached a threshold, it will stop and then being deposited. If you perturb the FGF signaling in this pathway, the pattern will change. Yeah.
Thanks. We have a question from Olya. In alpha E catenin depletion animals, do the adults display a phenotype of abnormal sensory organs?
That's a great question. I think the embryos looks fine. And the reason is because we are doing a tissue-specific depletion. If you are simply doing a mutation, they cannot survive. They cannot actually survive.
And a question from Sarah Paramore. Do you have any guesses as to what cue could be causing the cells at the back of the primordium to change their protrusion direction?
Yeah, that's really a great question. So we try to figure it out. Something we did is to manipulate the FGF signaling in this process. So as I said, FGF signaling is required for this neural mass to be deposited. If we take away the FGF receptors, and we can actually observe that collectively this class of cells cannot be efficiently deposited. They kind of being pulled away if we remove the receptors. However, this is not a cell autonomous behavior because when we're doing a transplantation of dominant FGF cells in the tissue, they just protrude fine. They just protrude fine, and also at some time they can protrude backward. So it's really not a cell autonomous behavior. It's in a collective way.
Great, thank you. And I think with that, if there's no more questions, we'll thank Weiyi once again. Oh, pardon, one more question. Mary Mullins asks, what happens to rosette when fewer cells are deposited as a neural mass? And does rosette formation fail in the primordium?
Yeah, that's a very great question. So in two situations, one is using Weiyi catenin depletion or the integrin beta 1 mutant, you can actually see that they can still form rosette, but you can see during this splitting process, the epiconstriction of the rosette is actually being teared apart. This becomes small clusters. This is the same for both Weiyi catenin depletion and also in the integrin beta 1b conditions.
Great, thank you. All right, if that's the last question, then once again, we'll thank Weiyi for a great talk.
Thank you.
And I'll pass it on to Olya to introduce our second speaker.
Hi, everybody. So my name is Olya Yarychkivska, and I'm happy to introduce our second speaker today, Robyn Allen. Robyn received an impressive dual degree, VMD, PhD, from University of Pennsylvania, where she worked under the guidance of Mary Mullins and Eileen Shore on F of P ACVR1 signals in the developing zebrafish. In 2021, she started her postdoctoral research at the University of Kentucky in the lab of Ashley Seifert, investigating organ size control and proliferative regulation during spiny mouse ear pinna regeneration. Robyn has a long-standing interest in regeneration since her undergraduate time, and her postdoctoral work has been recently published. Robyn has presented extensively at numerous international conferences and was recently elected the chair of the Gordon Research Seminar on Tissue Repair and Regeneration. Today, Robyn will tell us about fibroblasts from highly regenerative mammals and how they resist mechanical cues to drive regeneration. Robyn, the floor is yours.
Thank you, Olya, and thank you, SDB. I'm really excited to share my work looking at how spiny mice evade biomechanical cues to drive cell proliferation. So to start off, I'd like to introduce you to this charismatic creature, the spiny mouse, a rodent from the genus Acamys. And these guys are a really incredible model of mammalian tissue regeneration. They have the ability to regenerate skin and musculoskeletal tissues. And more recently, they've also been shown to have improved healing, functional recovery, and fibrotic resistance in multiple other organs following injury, including the spinal cord, heart, and kidney. So in the Seifert lab, we use the ear pinna as a model of complex tissue regeneration. You can see here in a regular mouse, when you create an excisional injury through the ear pinna, they will fail to restore any of that missing tissue mass and form scar tissue at the edges of the wound. In contrast, Acamys are actually able to fully restore all of the missing tissue mass and repattern it to its original architecture. And so, as you would expect, one of the primary places in which these species diverge from each other during the wound healing process is in cell proliferation. Both are able to activate a burst of cell proliferation following injury, but while in musk, cell proliferation precipitously drops after inflammatory resolution, Acamys is able to sustain consistent cell proliferation, in this case for weeks, until all of that missing tissue mass is restored and they finally downregulate it back to baseline. So the question I'm really interested in answering and what drives my research is, how does Acamys regulate tissue size? So how is it able to restore missing tissue without making too much tissue? And to investigate this, I started by looking at the role that biomechanics plays in this process. And I started by looking at dermal fibroblasts in vitro. So dermal fibroblasts are the primary cell that is going to proliferate and either generate scar tissue or generate a regeneration blastema in these species. And generally, when we think about the forces that they're experiencing in 2D cell culture, we focus on two primary forces, one being the stiffness of the substrate on which they reside, and the other being forces from cell-cell contact. So previous studies have shown that if you expose mouse fibroblasts to increasingly stiff substrates, they will undergo myofibroblast transformation. And this is a cell type that's commonly associated with fibrosis and ECM production, while Acamys in the same conditions will not undergo myofibroblast transformation. And in this study, they actually showed that there are no significant transcriptional changes in Acamys fibroblasts in response to their substrate, suggesting that they are insensitive to substrate stiffness. But I wanted to look at what happens with that other force that they're experiencing, which is cell-cell contact. Normally, cells, when they encounter each other, undergo a process called contact inhibition, which prevents them from migrating or proliferating as they perceive less space available on the plate. And interestingly, if you allow primary dermal fibroblasts from both mouse and Acamys to just grow on the plate until they stop proliferating, you can hopefully appreciate that Acamys are able to achieve much higher densities than musk cells, suggesting that they might also have reduced response to contact inhibition. And I've actually been able to look at this phenomenon in several species. So here I have two animals that are able to regenerate ear pinna tissue, spiny mouse and rabbit in blue and green, and then two animals that respond to injury with fibrosis, mouse and rat. And you can see that while these cells reach apparent confluency around the same time, if you look at the actual densities that they reach, my regenerative species are able to reach densities more than double that of what my fibrotic species can reach, showing that this is a phenomenon that seems to be conserved in highly regenerative mammals. So as a result, if you look at cell proliferation in these species, you can see, again, they reach confluence around the same time, but while mouse and rat will downregulate proliferation once they hit confluence, Achames and rabbit are able to continue proliferating into hyperconfluence and achieve these high densities. And importantly, the ability to reach these densities is not correlated to cell size. So even though Achames cells at subconfluence are significantly smaller than these other species, rabbit is about the same size as the other two, showing that this is not a cell size dependent phenomenon. And just to further emphasize this point, if you look at changes in cell size as density increases in both mouse and spiny mouse, you can see that these cells will reduce the area that they're occupying in order to accommodate each other, and these differences in cell size go away with increasing density. So based on this data, it appeared that my regenerative species have this ability to resist contact inhibition. And you can imagine how that might be important during regeneration when cells need to maintain proliferation and push a tissue front to fill a missing gap. And interestingly, if you look at densities across the regeneration that blasphema that forms as these cells proliferate, you can see here a cross-section of a regenerating ear with the initial injury site marked by this dotted line and the regeneration blasphema distal. In the periphery of this regeneration blasphema are alpha smooth muscle actin positive cells, likely myofibroblasts, that fibrotic associated cell type I mentioned before. And then deeper in the blasphema are these smooth muscle actin negative cells. And the smooth muscle actin negative cells are able to reach much higher densities than the smooth muscle actin positive cells, suggesting that density is also playing a role in cell types within the blasphema. So I really wanted to further investigate the interaction between cell density and cell phenotype in these species, and also how biomechanical cues might be informing this process. So I'll start by telling you more about the cell phenotypes that I can see. And there are two primary phenotypes that pop out when you allow these cells to hit confluency. So in mus, that alpha smooth muscle actin I mentioned before can either be globular and diffusely expressed throughout the cells, or polymerized into stress fibers, which mark our myofibroblasts, that fibrotic cell type I mentioned before. In Achames, however, there's actually a third phenotype that I see, and that is these perinuclear aggregations of globular smooth muscle actin. And if you look at the changes in polymerization and aggregation across these species, you can see in my scar-forming species, there's a fairly consistent maintenance of myofibroblasts, these polymerized smooth muscle actin cells, and very little generation of these aggregates. While in Achames, you get a huge increase in those perinuclear aggregates of smooth muscle actin as confluency is reached and beyond that. And this possibly allows them to resist the formation of that fibrotic phenotype and keep the myofibroblast transformation low. The second phenotype you can see pop out is that while fibroblasts from mouse are arranged in a fairly disordered fashion, the cytoskeletons of Achames cells actually appear to align with each other. So using orientation J, I can pseudocolor the actin cytoskeleton to look at the relative orientation of the fibers on the plate. And you can see that both species are randomly oriented at subconfluence. And Mus continues to be randomly oriented at confluence, but Acamese forms these big swaths of fibers that are moving in the same direction as indicated by this uniform color. And I can quantify the number of fibers that are within each of these orientations, and again show that Acamese is highly aligned with the majority of fibers falling within the same range of angles. And this phenotype actually will separate my regenerators from my non-regenerators. So mouse and rat have these randomly oriented cytoskeletons, while Acamese and rabbit have these highly aligned cytoskeletons. And again, I can quantify it to see that these two species keep their fibers in the same orientation. And this was very exciting because this is actually a phenotype that you see in some stem and progenitor cells with contractile ability. And it suggested that perhaps the fibroblasts from these regenerative species have phenotypes that are closer to stem-like or progenitor-like cells. So how are biomechanical cues influencing these phenotypes? To investigate that further, I first looked at YES-associated protein 1, which is a mechanosensitive transcriptional regulator that is downstream from several different mechanosensors. And importantly for my purposes, its activity is upregulated by a stiff environment and low cell density, and its activity is depressed by a soft environment and high cell density. Its activity is also controlled by a subcellular localization. So phosphorylation of YAP will cause it to be exported to the cytoplasm where it is inactive, and dephosphorylation will allow it to move into the nucleus where it can activate various transcriptional cassettes, including those that promote proliferation and fibrosis. And YAP has been identified as a promising target in skin healing in mouse dorsal skin wounds. Inhibiting YAP will allow hair follicle neogenesis to occur, while in Acamese ear regeneration, YAP inhibition will actually delay ear closure. So when I look at the subcellular localization of YAP to get an idea of its activity across these species, what I see is that there is not actually a strong correlation between the proportion of nuclear YAP and whether or not these animals are able to regenerate. I do, however, see that in my spiny mouse, I have a significantly higher proportion of nuclear YAP relative to total YAP compared to my other species, suggesting that its baseline activity is higher than these other species. A better way to look at YAP activity, however, is to look at its direct transcriptional target. So here I have a list of genes that are known to be very highly correlated in their expression to YAP activity, and I've split them roughly into cells that are pro-proliferative genes that are pro-proliferative and genes that induce cell cycle arrest. And you can see there's actually a lot of differentially expressed genes on this list between Mus and Acamese, but there's not a bias towards proliferation or cell cycle arrest between these two species. However, when I look at changes in expression in response to increasing density, some interesting patterns emerge. So when I look at my pro-proliferative genes, Acamese is actually able to maintain most of these genes at a stable expression with increasing density, while Mus begins to downregulate a good proportion of these genes. And this again suggests that Acamese has resistance to contact inhibition, allowing it to maintain cell proliferation at higher densities. And then when I look over at my cell cycle arrest genes, I also see upregulation of a couple of them in Mus, but not much change in Acamese. So I wanted to look at what happens when I manipulate YAP activity. There is a role for YAP in both proliferation and fibrosis. So what happens if I kick it up in both species that seem to have differential activation of its gene target? And to do this, I used a small molecule inhibitor called Truly, which prevents phosphorylation of YAP and retains it in the nucleus where it's more active. And I can see that when I treat these cells of both species with really high levels of this small molecule inhibitor, I can very robustly induce myofibroblast transformation, creating these big cells with abundant smooth muscle actin positive stress fibers. However, because this is a very stressful thing, this will arrest cell proliferation, and I can't look at what happens at these high confluences. So I use a lower concentration that still allows cell proliferation. And what I see is there's still a trend towards increased myofibroblast transformation in both species, with a significant increase at subconfluence in Acamese, suggesting that these fibrotic pathways are being upregulated. In addition, I see that while, like I mentioned before, we get these smooth muscle actin aggregations in Acamese at confluence, these are almost completely lost when YAP is activated, suggesting that it plays a role in balancing the localization of smooth muscle actin in these cells. And lastly, when I look at the ability of Acamese cells to align with each other, I see that YAP activation basically ablates the ability. Some of the cells still are able to align with each other in small clusters, but they are unable to form those big aligned sheets across the entire area of interest. And importantly, like I mentioned before, this dose of YAP still allows these cells to reach the same very high densities, indicating that the ability of these cells to reach high cell densities is not dependent on the aggregation of smooth muscle actin or the ability to align their cells, and that rather these phenotypes are probably a product of reaching those densities to begin with. So the last experiment I wanted to show you is what happens when you wash out this small molecule inhibitor. Can any of these phenotypes recover? So in this experiment, I treated my cells with my YAP activator at subconfluence, washed it out, and then allowed them to grow to confluence before collecting. And what I found is that while there's not much change in the number of myofibroblasts at confluence, I do see recovery of those smooth muscle actin aggregations in my Acamese cells, suggesting that this is a dynamic process. On the other hand, when I look at the ability of these cells to align, even though they had the YAP activator removed at subconfluence, once they got to confluence, they still were not able to align across the area of interest, indicating that some kind of irreversible change had taken place in these cells. So to conclude this talk, I want to look at this phenomenon through a lens of YAP. And what seems to be happening in mouse is that normally you have robust response to contact inhibition and sensitivity to substrate, which allows for the activation of fibrotic targets while repressing proliferative targets once you reach high density. On the other hand, Acamese seems to have a weaker response to contact inhibition and possibly no response at all to their substrate, allowing them to keep activation of fibrotic targets low while keeping proliferative targets highly expressed at high densities. And what I'd like to do in the future is investigate how these phenotypes and phenomena are playing a role in vivo to allow regeneration to take place in these species. So with that, I'd like to thank my funding sources and my lab and the mice, and I'll take any questions. Thank you.
Really interesting talk, Robyn. Thank you so much. Please put your questions into the Q&A. We already have one question from Carrie Adler. Beautiful work, Robyn. In the confluent Acumis cells, do the alpha smooth muscle aggregates persist, or are they eventually degraded? Have you seen them in vivo?
Yeah, so it seems like once they form, unless you activate that YAP, they stick around. I've gone out to about a week, so allowing the cells to sit at hyperconfluence for 4 days, and they still continue to be there at that point. As far as whether they're in vivo, I'm not sure yet. We use paraffinization for most of our tissue preps, and that unfortunately destroys the cytoskeleton. So I'm working on getting a better look at what's happening with those fibers in vivo.
We have another question from Jennifer Mitchell. This is very nice. Learning from Acumis experiments and how they respond to YAP activation. Have you tried to do the opposite in mouse cells to achieve the wild-type Acumis traits that seem to be linked to the regenerative potential?
So yeah, I didn't say it, but you might have noticed in my graph that activating YAP doesn't really allow mus to achieve the same densities. So there's definitely other factors at play. When I look at inhibiting YAP, you can reduce the fibrotic phenotype in mus and reduce the number of myofibroblasts they're forming, but it does also inhibit proliferation. So there's definitely some downstream tweaking that needs to happen. And I think that that differential activation of YAP targets makes a big difference between the species.
We have another question from Gage Crump. Great talk. What is known about the interaction between myofibroblasts and the cells that regenerate the chondrocytes? Do they derive from the same mesenchymal precursor, or do they signal to each other?
Oh, that's a really interesting question. Something that we're in the process of working on. Some work by one of our graduate students shows that there's 3 main cell types that we're able to identify within the blastema, and one of them is a SOX9 progenitor, which we believe gives rise to the chondroblast. And the other is CRAB-P1 positive cells, which some of you might recognize as a pro-regenerative phenotype for fibroblasts. And each of these cell types appears to occur in distinct areas within the blastema. They're pretty separated from each other. So there's likely some crosstalk between them, but we haven't discovered yet what's going on. I think the myofibroblasts are kind of required at that tissue front, but they need to go away in order for the other cells to come in and take over.
I would like to ask a question, if you can tell us a little bit more generally about, that seems that these cells divide so much more. So do you know anything about the cancer potential in these cells? How long do these species live? I'm thinking about naked mole rats and how they live so long compared to their size, but they never get cancer. What happens with these Acumis mice?
Yeah, I love that question. I think about it a lot. Let me start from, there is a theory in regeneration that there is kind of a balance that needs to be played between proliferation and the potential for oncogenesis. But there's been some work that suggests that regenerative species might have resistance to cancer. Certainly in the axolotl, you can see them form tumors, but they tend to be more benign. In Acumis, we see them form tumors in response to what we believe is a papillomavirus they can get on their ears. So they do undergo uncontrolled cell proliferation, but it seems to be fairly well controlled. And there's actually some papers coming out soon looking at if you can induce cancer. I forget what drug was used, but in the dorsal skin, and the Acumis seem to be really, really resistant to it, while black 6 mice will form numerous masses. Acumis do not. However, sorry, there's more to this story. We can see that in some of our older animals, and especially if they're overweight, Acumis can actually spontaneously develop diabetes. And in some of those animals that we believe to be diabetic, you can see overgrowth of tissue after a ear punch. So that hopefully answers your question. It's a very complicated phenomenon, but maybe resistant, but not completely.
Well, thank you. It's super interesting how these non-model organisms could inform us about all of these human conditions potentially. So thank you so much, Robyn. We don't have any more questions right now. So I would like to close our session by thanking both of our speakers, Weiyi and Robyn. Those were excellent talks. And this seminar has been recorded, and the video will be available on the SDB website next week. And please join us for the next month's seminar on Friday, January 9th, 2026, where Priyom Adhapok from Duke University and Zongyuan Liu from University of Michigan will present their work. And thank you all for coming today.